This is a video in which we'll have a look at some of the key features of the structure of DNA, which you'll need to know for your chemistry A-level. Obviously, it's important just to be aware of the significance of DNA, and what we know is that it carries information, we call it genetic information, which cells use to direct the formation of particular proteins within the cell, which gives the cell its characteristics. And we'll look at the way in which DNA directs the formation of proteins in a second video. Okay, so in terms of the chemistry, we first of all need to be aware of how the molecule DNA is formed by condensation reactions from the monomers that make up DNA, which are known as nucleotides. And we'll need to look in a bit more detail at the actual structure of the nucleotide monomer. So just giving you an overview of the structure of DNA, and we will come back to this overall structure in more detail later on. We need to be aware of the fact that DNA is a very long molecule. It's a biological polymer. We can think of it as containing these two chains or strands. Each of these strands is a long polymer chain, and they're twisted around one another in what we know as a double helix. So just like we're expected to know that proteins are polymers made up of amino acids, we're expected to know the name of the monomer from which DNA is made up. And DNA, and indeed its sister molecule RNA, are made up from monomers known as nucleotides. Nucleotides. Now, it's not quite as simple as that, because actually each nucleotide contains three components, and those three components attach together to form a single entity known as a nucleotide, and it's very important that you know what those three components are. Luckily, there's information on the datasheet to help you. So at this stage, you might like to make sure that you've got a datasheet available to look at, and we'll hopefully allow you to see how you can use it to remember the types of structures which form the components of a nucleotide. So pause the video while you find the datasheet, and can you make sure that you find the page on the datasheet? It's page 3 that shows you these structures here described as the monomers of DNA and RNA. Okay, what are those three structures? Well, they represent the three components of a nucleotide. A phosphate group, and you can see that's labelled here. A sugar molecule, and there are two sugar molecules here, one called ribose, one called deoxyribose. One's found in RNA, one's found in DNA. And then a base, and there's 4 bases here, and you can see the names that are given to them. Now, technically speaking, these are the bases found in the molecule RNA. DNA contains a slightly different base there, known as thymine, but you'll see on your datasheet thymine is mentioned, and it explains that it actually is virtually identical to uracil, but with a CH3 group there. Okay, so we should be able to link the diagrams we can see in the datasheet here to the memory of the three components of a nucleotide. So we've mentioned already that in DNA the sugar is called deoxyribose. You can see that this sugar molecule here has a 5-membered ring. If you compare it with more familiar sugars like glucose, which have 6 atoms in a ring, it obviously is slightly different in structure, but it has the similarity of having several OH groups attached, much like in glucose. You'll notice that ribose has got 1 extra OH group there. And then we talked about the bases present, and although the bases are shown for RNA, we're expected to know the bases present in DNA. Now, you can use the names. You can often get away with just letters to represent those names, A, T, C, and G. And there's the thymine molecule with the extra CH3 group there. So hopefully, faced with any of those structures in a question, you'd be able to identify them from the datasheet. Similarly, you'd be able to use the datasheet to actually draw out any particular structure that was required in a question. And it turns out that that is indeed what we are required to do. We're required to actually potentially draw out the structures of those molecules and show how they can actually join together by condensation reactions, in other words, reactions that release a small molecule, in this case water, to form the chains that make up DNA and also the bases that are attached to the chains, and we'll see later on the significance of the bases that stick out from the chains in DNA. So these are the two learning outcomes that will follow in the next part of this video. So we'll actually look first of all at how a sugar molecule can join to a base. And they can obviously select any pair of sugar and base they want, and ask you to be able to show how that reaction happens. So here, for example, we're going to take that molecule there, which is deoxyribose, and we're going to look at a thymine molecule and think about how the thymine molecule is going to join to deoxyribose. Well, luckily, the way that it's drawn out in the data sheet, it's very easy to see how that reaction happens. If you imagine bringing down the thymine molecule until it's next door to the deoxyribose, you can almost see the way in which the water molecule is going to be lost. So you might like to pause the video while you actually think about how you would draw out the structure formed from deoxyribose reacting with thymine. And yes, we end up getting a bond formed between the carbon atom of the deoxyribose and the nitrogen of thymine, and a water is lost. And so let's have a look at another pair there. So again, we take our deoxyribose sugar, we bring down cytosine. So again, pause the video while you think about what happens when those two molecules join together. And yes, there we are, the carbon and nitrogen bond together with the loss of water. Exactly the same thing happens with the final two bases, adenine and guanine. You'll notice here that the molecules are slightly more complicated. It's not immediately so obvious which nitrogen atom, for example, will be involved in bonding to the deoxyribose, but look at the help they've given you. They've actually shown this nitrogen-hydrogen bond quite explicitly on the data sheet, and that's your clue that that's the atom involved in forming the water molecule, and hence the bond that links the two molecules together. So let's look at what happens when those two form bonds to the sugar molecule. Deoxyribose, bring down an adenine, form the link, lose water. Deoxyribose, bring down guanine, form the link between the two, lose water. Now that sugar-base combination is actually known as a nucleoside, and nucleosides can then join to one another by using phosphate groups to link them together. So we'll see how the sugar molecule of that nucleoside is actually then used to bond to a phosphate group. So here is a nucleoside consisting of a cytosine molecule and a deoxyribose bonded together. Let's bring down a phosphate group. And again, you'll notice that the way that the molecules are drawn out is extremely helpful. If we put a phosphate group on top of a deoxyribose, you can see these two OH groups nestling up quite close together. So you might like to think now about what happens when a bond forms between a phosphate group and a deoxyribose in a condensation reaction. So pause the video while you give that some thought. So we lose a water molecule, and now the oxygen atom from the phosphate ends up bonding to the carbon. Notice the carbon now is not bonding directly to the phosphorus, it bonds via an oxygen atom there. Now what we've got is what's called a DNA nucleotide. What you need to be aware of is that that nucleotide is now able to form bonds to other nucleotides. How can that happen? Because there's an OH group on that phosphate group there. There's an OH group on the deoxyribose, so you could imagine placing another nucleotide on top, and that OH group on the phosphate and the OH on the deoxyribose are now able to continue the chain in both directions. And so we end up getting a sequence of sugars, phosphates, along a long line with bases sticking out at right angles, and that's the key to your DNA structure. So we quite often represent a nucleotides much more simply by showing the sugar, the phosphate and the base. You can imagine another phosphate coming along here and bonding to the sugar, another sugar molecule coming along and bonding to the phosphate. So there's various animations that we can show that happening. So here's a nucleotide, here's another representation of a nucleotide, a little bit more detail there, and we can show them now joining to produce this DNA strand. So again, watch for the atoms on the phosphate and on the deoxyribose sugar here. So along comes a second nucleotide and a third nucleotide, and here are the condensation reactions causing them to bond together. Now, technically, you might well be required, given a diagram showing one nucleotide, to show a second nucleotide bonding on top. So you should get used to the idea of drawing the OH of one ribose sugar immediately adjacent to the phosphate group and figuring out how a condensation reaction can occur between the two. So what we have here is a sugar phosphate backbone. That's the strand of DNA that we saw in that original diagram, and notice there's a base sticking out from the sugar molecule. It's important to remember that the base is specifically bonded to the sugar and not to the phosphate. Lots of ways in which that might be represented in textbooks or exam questions. We could get the level of detail we show here, or sometimes you can represent the sugar phosphate backbone simply by a long chain here with the bases sticking out at right angles, similar to how we showed the amino acid chain here with the peptide links joining the amino acids and the side groups. In this case, we've got a sugar phosphate backbone and the bases sticking out at right angles. Important not to get proteins and DNA confused. There are similarities between the two, and there's clearly a relationship between the two, but they have very different structures. And then sometimes, because actually the important thing in DNA is the sequence of bases, sometimes DNA is literally shown just as a sequence of bases, and you have to imagine that actually there's a sugar phosphate backbone strand attached to that. So that concludes the first video looking at DNA structure. In the second video, we'll look at the way in which the bases pair together and get a sense of the overall structure of DNA and the features that we will need to recall from that.